tape in, I'll interject here instead of typing. You're unmuted now, but we're not hearing you yet.
I don't know why you can't hear me. Everything was working.
We were hearing you great, man.
Hearing you for briefly, yeah.
It's so weird.
Yeah, we can hear you fine, man.
You can? Oh,
yeah.
All right. Everyone's just staring into the void. All right. So you hear me fine?
Yeah.
So okay. And the presentation looks fine?
Go ahead and pull it up, and then just make sure it goes in the presentation mode, and you can go from your title, your first slide, and back, just to make sure it looks good.
All right. So when I started sharing last time, I didn't hear anybody, so I was worried that I somehow got cut off.
Gotcha. No, you're sounding good so far.
All right.
Okay, Matt, I'm going to introduce.
All right. So you should see now just a full slide, yes?
You got it.
Yes.
Yep.
Perfect. Okay.
Lauren, give me a second. I'll highlight you, and we'll get started.
All right. Well, hello, and welcome everyone back to Science in the Age of COVID-19. Today, we are super excited to welcome back Ben tenOever, a professor of medicine and microbiology at the Icahn School of Medicine in Mount Sinai in New York. Ben is an absolute rock star in virology, so much so that he was one of the first guests in our seminar series on May 7th. In that talk, he talked mostly about the host response, the host innate immune response with interferons and downstream signaling mechanisms to SARS-CoV-2 infection. His lab works on a lot of other things and as well, and he's going to update us on a number of other stories about SARS-CoV-2 today. So Ben, take it away.
All right, I will. Just assume you can hear me okay. Sorry, just doing some last minute adjustments. I'm not in the dark. All right, I didn't even change the title. I'm sorry. I was literally working on this slide until just 5 minutes before this. But that was in the hopes to talk about some new data that I haven't been able to discuss until now. As of yesterday, SARS-CoV-2 has surpassed 60 million cases worldwide with almost 1.5 million deaths. I don't think it's a surprise to anybody that this is an important problem that needs to be addressed. And I am optimistic that the onslaught of vaccines will come online soon. But in the meantime, today I'm going to talk mostly about just the biology of this virus, why it does give the heterogeneity that we see in people, at least what our data would suggest might be the underlying cause of that, and to discuss at the very end the use of interferon as a prophylactic, which has been a common theme here in the lab. So this is the virus that we're going to talk about today. It's obviously a beta coronavirus. I like to show this figure only to appreciate that it is a much more complex virus than say influenza with different segments. But despite the fact that it's a single-stranded, very long piece of RNA, it does go through some viral acrobatics, which contribute to, I think, a lot of its pathology, as I hope to convince you today. So ORF1A and ORF1B is the replication machinery. They get cut up into smaller pieces that each have their own unique function. And I don't think that these are actually the major driver of disease in the host cell, but rather at the 3' end of the genome are a number of accessory proteins and structural proteins, and they get transcribed in a very unusual way where the antigenome essentially gets produced moving in this direction. And when it hits one of these transcriptional regulatory sequences or TRSs, it essentially jumps template to recreate the 5' end of the genome. And then those subgenomic RNAs can be used to make mRNA for all the various structural and accessory proteins. But I think it's probably this jumping and this movement of the polymerase from across segments that generates a lot of RNA that would be aberrant or viewed as foreign by the cell, and therefore initiate an antiviral response to that. And just as evidence of that, this is unpublished data. In fact, a lot of what I'll talk about today is unpublished data. But this is an antibody, what you see in green there, to the general structure of double-stranded RNA. So it's not specific to a particular sequence, but you can appreciate that even in cancer cells, these are a long carcinoma cell line called A549s that have been engineered to express ACE2. at baseline, you see no green signal there, suggesting that cells, in their wild type state aren't generating double-stranded RNA. But if you infect these same cells with SARS-CoV-2, you now see that green becomes a very prominent signal in the cell culture, suggesting that this virus is in fact making a lot of an RNA that the cell would view as inflammatory. And so this is kind of an overview of what I've been studying for 20 years, what my lab is interested in studying, what many other labs work on, and that is how cells respond to virus infection in general. And I don't want to get into it in great detail just yet, but the big take-home message here is that things like double-stranded RNA are in fact detected by the cell, and they elicit a pretty well-known signal transduction event that ultimately leads to the activation of these two families of transcription factors, notably IRF3 and the NFkB family, which is usually P50, P65. And they make this giant complex termed the enhanceosome and are responsible for the induction and really pulling the trigger on the antiviral response through the production of either type 1 or type 3 interferons, which are secreted proteins, which are depicted here on the right, which induce their own signal transduction through cells, which can work both autocrine, so in the very cell that was infected, but more so in the cells in the neighboring area, where they can fortify and induce antiviral defenses through a complex called interferon-stimulated gene factor 3, or STAT1, STAT2, and another IRF member called IRF9. And so the even higher level view of this would be something along these lines, where basically you make an aberrant RNA that the cell recognizes is not from itself. It induces the dimerization or oligomerization of various pattern recognition receptors that then activate transcription factors that induce this antiviral response. And the response comes in these two flavors. One is a call to arms. This would be the interferons and the interferon-stimulated genes, which are going to serve to directly slow virus replication and try and inhibit it as best it can. And sometimes this arm alone can be successful, but usually this is not enough, and you need a much more sophisticated adaptive response, and that comes in the form of this call for reinforcements, which is mediated by a chemokine trail, which is secreted proteins that are going to create a gradient to tell neutrophils, monocytes, T cells, and B cells to come to the site of infection and learn to recognize the pathogen for exactly what it is, and then mount a lifelong response against it. And so in this lab, we do a lot of sequencing. And so these are early days. In fact, this is one of the slides I presented way back in May for this same seminar series. But I still really like this figure because it shows some really important data. So one, what you're really looking at here is a histogram. So you're just looking at the number of reads that map to the genome, which is here on the axis. And one thing to appreciate is that in the absence of ACE2, so when these A549 cells are just expressing mCherry, you get very, very little replication, if any, of RNA. But if you simply add ACE2, you can see you go up by many orders of magnitude, and you now have 1,000-fold coverage across the genome and then some. And interestingly, regardless of whether or not you come in here at a low MOI or a high MOI, essentially you saturate out at this very high level of RNA, and you're going to see why that happens in just a minute. And interestingly, and again, I talked about this last time, so I don't want to stay on this topic too long, but it is very interesting in that if you look at the differentially expressed genes in these high throughput sequencing efforts, there is a stark difference between coming in at a low MOI of, say, 1 virus per 5 cells versus, say, 2 viruses for every cell. And so just as a reminder, this is not due to increased levels of virus, because the levels of virus seem to be saturated based on viral reads. So they are at their maximum capacity, and yet the host response to these two conditions is very, very different. And the stark difference here is really outlined here in this blue box. And these are the type 1 and type 3 interferons, that call to arms that I mentioned a few slides ago. And so that's the direct interferons themselves. And then this large white chunk and this lighter chunk here, these are all the genes that are induced in response to these interferons. So really the difference between low MOI and high MOI is the difference between interferon being disabled or being enabled. And interestingly, on the right side of this graph, I'd like to depict is the fact that regardless of MOI, we see the call for reinforcements in both conditions. And I hope to explain this to you momentarily as well. And so the first thought, and this is going to be our first new section, is, does MOI matter because of timing, which it certainly may play a role, or is it actually have more to do with the fact that where this gene, where these gene signatures are coming from. Is it that they're coming from infected cells versus uninfected cells, and therefore a low MOI has more cells that are uninfected and capable of signaling? And the reason we came to this question was largely because when we started doing this RNA sequencing itself, one of the things that became very obvious was you could tell if you ran, say, 12 samples on a flow cell on a NFkB run, and let's say they were uninfected and infected with SARS-CoV-2, on your initial bow tie alignment where you grab each read and you try and determine how many genes match to each particular transcript, you usually get numbers of like 95% of all the reads generated map to the human genome or the hamster genome or the ferret genome or whatever kind of tissue you're working with. But in SARS-CoV-2 infected cells, you could tell right from the first alignment that they were infected because that rate of alignment of 95% plummeted down to sometimes as low as 20% or 30% or 40% reads, suggesting that the other remaining RNA was in fact viral in nature. And so this is just a simple bar graph to show you how high the poly-A mRNA population can be with regards to being viral RNA of origin. So you can see here at 24 hours in A549 cells expressing ACE2, we can get as high as 80% of all the reads are virus in origin and not of host origin, suggesting that any kind of transcriptional signature that's coming from this is probably not coming from the virus-infected cell because most of that RNA is either gone or being destroyed. And just, to show this another way, this is single cell data. And instead of, I like this figure because it demonstrates that out of the, 40,000 unique transcripts picked from mock infected cells, these have no SARS-CoV-2 reads. So these are, we are assuming bystander cells in a population. When you infect these cells, you can see this population of yellow, that is the now growing virus. But it's not really that the virus has taken over 80% of the mRNA population. What's actually happening is the virus is accumulating to levels that it probably would compare to most RNA viruses. But at that same time, it is destroying all of the host RNA so that what's left is mostly viral RNA. And this is very clear when we go to single cell resolution. But interestingly, when we look at the differentially expressed genes, which requires a little bit of a lot of statistics actually to account for the change in actual numbers of genes, what you can appreciate is that there is some signaling coming directly from the virus-infected cells themselves. And these produce a very, very strong NFkB signature. So we've just outlined a few genes here, but the number one annotation of all these genes are NFkB-driven genes, which is quite interesting because it would suggest that the virus is essentially taking over the cell, but for reasons that I won't be able to explain to you today, NFkB is left largely on and untouched. And this is further supported by work done by a really talented student here named Skylar Yule, who did ATAC-Seq on these same cell cultures and found that there's this massive enrichment. So the first 1, 2, 3, 4, 5, 6 peaks that really shadow all other peaks on an ATAC-Seq data. And for those of you who don't know what ATAC-Seq data is, it's really measuring the accessibility of DNA promoter elements. So if a gene turns on, generally the promoter area will open up its histones to allow the transcription factors to be able to engage in a sequence-specific manner. And so this is basically measuring accessibility in response to virus. And so the fact that NFkB is showing up in all these top peaks is really just further confirmation that virus infection is leading to a very strong activation of NFkB in the complete absence of many other transcription factors that are known to be induced by virus, including IRF3 and IRF7, which are key pivotal roles in that initial induction of interferon. So even if you didn't get all of those details, what this does really clearly show is that even though the cell has all of these mechanisms in place to combat virus infection, we know that antagonists, like the Cato lab described initially of ORF3b or the protease, the PLP protease, are likely blocking only the IRF3 arm of this antiviral pathway while leaving NFkB untouched. And of course, the virus also has additional strategies. So NSP1 has been well studied and mapped and crystallized to bind to the 40S ribosomal subunit. So this is certainly a major cause of blocking translation of host genes. It's unclear at this point whether or not it's also involved in some of the degradation of RNA. And then it also has this other open reading frame, ORF6, which blocks the nuclear translocation of ISGF3. So the virus is really having its way with a cell, but for whatever reason, NFkB, like I said, is left on. And this really does create a unique phenotype, which we viewed in both the ATAC-Seq data and in the single cell data, where virus produces double-stranded RNA, that RNA gets recognized, and then there's a response to it. But the vast majority of that response is then blocked by virus. And so you don't get the call to arms, at least from the infected cells directly. So you don't get interferons, and any ISGs that would have been signaling from interferon from other cells would be blocked by things like ORF6, which are going to prevent the translocation of ISGF3 into the nucleus. However, at the same time all of this is going on, as I said, NFkB is left on. And NFkB is largely involved in the production of both apoptotic genes. NFkB can actually both be apoptotic and pro-survival, depending on the context of activation. But it is also very much involved in the production of chemokines and pro-inflammatory cytokines, which are the hallmark of COVID. So, taken again at this high level, it's already painting a picture that virus infection is going to lead to a very strong call for reinforcements with a very diminished interferon response with regards to direct antiviral factors. And so the other thing, though, that's noteworthy here is that in addition to this, we know that SARS-CoV-2 induces a massive apoptosis of infected cells. So when we do these infections in A549 cells, as you can imagine, any virus, any cell that is now 80% viral RNA is not going to live very long. And indeed, we do see complete culling of the cells in a dish. And this is going to be something I'm going to return to numerous times in this talk. But I just want to throw this out there right now so that it's in everybody's head that, while this cell is dying, and again, this gets back to our low MOI versus high MOI. So in a low MOI condition, when this cell dies, it's possible that this neighboring cell has not yet been infected. So even if the virus completely shuts down the RNA, the translation, and the detectability of a virus here, the material that's going to bleb off these cells, and it's been shown that SARS-CoV-2 induces both pyroapoptosis, apoptosis, and necroapoptosis. These blebbing bodies could in fact turn out to be the PAMPs for neighboring cells that are uninfected. And of course, in these cells, they're going to endocytose or bring in the inflammatory RNA or the PAMPs, but now you don't have the viral proteins to antagonize it. So the interferon that you can observe, say, at a low MOI infection or in vivo, is likely coming from these neighboring uninfected cells where they have a chance to induce some interferon. But of course, even on a larger context, this still will never be what it should be because you're losing all of that signal from the infection. But just to bang this point home, I'm not going to talk a lot about this paper either because it's already published, so you can just read it. But the take home here is that we, in collaboration with Neville Sanjana at the New York Genome Center, used this extensive cell death to, in fact, perform what turned out to be a very rapid screen to identify essential host factors required for SARS-CoV-2 infection. And so, as you can imagine, you can easily take advantage of this by adding in these CRISPR libraries. And this is Neville's playground. He is his whole lab was just incredibly talented at generating a comprehensive 6 guides per every gene knockout library in A549 cells. And we simply did 2 different MOIs. This was the work of a very talented postdoc named Tristan Jordan, who is on the job market if anybody's interested. And Tristan simply did an infection at 2 different MOIs and then allowed those cells to just to die in response to SARS-CoV-2 and collected the remaining DNA that was left in any surviving cells to determine what guides were still represented in that space. And like I said, I don't really want to go over this in great detail, but this work very successfully and very cleanly, I think, pulled out all of the requirements for the ability of this virus to enter cells. So Tristan and Jarko, the 2 first authors of this paper, quickly demonstrated all of the machinery required for ACE2 expression on the surface, the endocytosis of the virus, the trafficking of the virus once in the cell. And these all came out very clearly in subsequent follow-up screens, either by siRNAs, by other single CRISPR screens, or even by small drug inhibitors. Really just going to show that once the virus gets into the cell, it seems like it's the end game. So if you can prevent viral entry, you can save your cells. But once the virus is in, even if you knock out some essential factors, it seems like death still wins out. And this may have to do with some of the cytokines that are being released by that NFkB signature, such as TNF-alpha. And so this is all, of course, well and nice. It's all cell culture. It tells us a lot about the initial biology, about why we see so many chemokines and why we see a disproportionate amount of pro-inflammatory cytokines when we don't see or a low amount of interferon. And so the next obvious question, well, does this extrapolate out to much more complex tissues like an actual in vivo experiment in the lung? And I'm not going to talk about any human data today. I'm going to, in fact, just talk about the hamster model for this, which I believe has really shown itself to be the best animal model for, or at least the best small animal model for SARS-CoV-2 infection. And I say this because unlike mice, hamsters are naturally acceptable to this virus. So we don't have to change the virus or the mouse to achieve a disease phenotype. Like people, male hamsters are more susceptible to morbidity than female hamsters are in response to SARS-CoV-2. And it's been demonstrated that there's an age-dependent progression of infection in hamsters as well. So it really does seem to be the best model we have with regards to being able to model how this disease manifests itself in vivo. And so we initially actually started with the ferret model. So I would have talked back in March about the ferret model. And the ferret model was okay. You do get replication. It is a productive infection. They do transmit the virus. But for reasons that I can't explain, the virus, the SARS-CoV-2 virus in ferrets seems to be very much confined to the upper respiratory tract. So in the nasal turbinates, we see a lot of virus. As you go back into the trachea, you see less virus. And when you get down into the lung, really there's no virus there at all. And I'd be happy to talk about a discussion as to why that might be the case for ferrets, but it's certainly not the case for hamsters, which is another reason why we think hamsters are probably the best small animal model. And so all you're looking at here is just simply two different MOIs, a low MOI, which is just 100 plaque-forming units, so 100 virions capable of initiating infection, versus a much higher dose of 10 to the 5 PFU. And I just wanted to show this. We basically give the animal xylene and ketamine, so they are unconscious. We put them on a heated mat, and we simply add 20 microliters of virus into their nose, and then allow them to naturally wake up on this warmed bedding, so that they have the most amount of comfort possible and then recover rapidly thereafter. And one of the things that we liked about this result was that while most people in the published world were using much higher titers like 10 to the 5 PFU per ml, with the help of Yoshikawa Oka, we tried as little as 1 and 10 PFU, and these are equally effective at creating the same disease phenotype, but we believe represent a more physiological setting for the virus. And so we settled in on 100 PFU, mostly because at 100 PFU, you get 100% transfection of the cohort that you infected, but it's still at a level where you can see that the virus climbs with time. So unlike high MOI, where the virus is high and stays high until day 14, at 100 PFU, you see the virus comes up, it peaks here at day 6 and then falls all the way to day 14. And so this is going to be the model we're going to follow throughout, although there will be some high MOI data as well. And so as I was saying, one of the nice things about the hamster model is that they do show some levels of morbidity. So they, when we get them, we use 4 to 6 week old hamsters. These are probably representing the teenage world out there. And they are still growing. So when we get them, they eat rapidly, and as you can see here, given a vehicle like PBS, they gain weight over a 2-week time frame. But when given SARS-CoV-2, you see that weight drops or at least plateaus, suggesting that these animals are not feeling great and are not drinking as much water or eating as much food, and therefore not gaining the same amount of weight. However, you'll find that once they do recover and clear virus, they do start gaining weight again. But overall, you can tell that this virus is having an impact on the overall physiology of the animal. Moreover, if you start examining some of the tissues, so this is a trachea, and I don't know if it's going to come out very well in your slides, but some of the basic morphological changes that you can see is a complete loss of cilia, which have been reported by others. And so this virus is clearly getting into the upper respiratory tract, and it's climbing its way down, or making its way down into the lower respiratory tract, and it's doing damage basically along the way as it goes. If you take lungs on different kind of longitudinal studies, so it's the same 2-week time course, but now we're taking lung histology. So the blue, you're seeing nuclei, and then the brown, you're seeing the staining of the nucleoprotein. So you're looking at SARS. And so again, at 100 PFU, you can see that on day 2, you can start seeing that the virus is taking hold in the lower respiratory tract, but it really does peak here at day 4. We see, very robust staining in the alveolar space. That then dissipates more on day 6 and 8 before eventually it gets cleared. However, it's interesting that on day 14, it is still not difficult to find cells staining for N protein. And again, I will talk more about this later, but it's interesting to note that if you were to try and do plaque assays on day 14, you won't find a single plaque forming unit. If you homogenize the lung and PBS and put them onto susceptible vero cells, you won't find any death of the vero cells, yet you do find staining of N, and you can pick up N by PCR, suggesting that maybe not replication competent products, but essentially viral debris or DI particles does hang around for a long time in various tissues in the animal, which would also help explain why numerous people can still test PCR positive even once they've seroconverted to IgG. So in addition to this, we also see lung infiltration, of course. So in a SARS-CoV-2 infected hamster, you can certainly appreciate that you get a very large infiltration of cells. These are mostly neutrophils. So it's interesting because usually with viruses, you see an infiltration of monocytes first, but with SARS-CoV-2, we see more of a bacterial infiltration-like system where we see heavy neutrophils. And there's a number of things here I want to share. I'm not a pathologist, but we did have a very much a board certified pathologist go through all of these slides blindly. And the first thing I want to show you here are things like this. So this is a neutrophil dying. So she refers to this as a chocolate chip cookie, but the idea is that these small blebbing bodies are all apoptotic bodies from dying monocytes or even dying pneumocytes that are infected by SARS-CoV-2. And again, we're going to get back to this in a moment, but clearly there is a lot of apoptosis in the lung itself. The other thing we see is what was referred to as neutrophil nests. So these are neutrophils upon neutrophils. So what you should really see is something more like this, where a single neutrophil is coming to the site of infection. It's seeing that chemokine gradient, that call for reinforcements we talked about, and it's coming to the site of infection in order to be activated and induce some kind of inflammatory response. In terms of neutrophils, that this response generally comes in the form of nets. So neutrophils actually expel out their genomic contents and create essentially a net of genomic material, which has been thought and proposed to be involved in the harvesting or trapping of bacteria. However, of course, for virus infection, this is less helpful, but this is the response that's being elicited in following SARS-CoV-2 infection. Again, largely because we do see an interferon gamma signature and more of a response that would be more thought of as bacterial and antibacterial in nature rather than antiviral in nature. In addition to this, we also see this very strange phenomenon of edema. And you can see this everywhere. In fact, in some of the older slides, you can see it as well. And it's the reason we chose this slide to show is because this is surrounded by actually healthy tissue. You don't see a lot of SARS staining here. We don't see a lot of neutrophils here. And yet these blood vessels are leaking. So I don't know if it comes out well on here, but basically what's happening is the blood vessels that are intercalating through the lungs are suffering from leakage. So you get leakage, which would allow for both blood to come into the lung, and we definitely see hemagglutinin in the lung. Hemoglobin in the lung, but it also would allow material to go into the bloodstream. And again, this is a point I would like to make now, but come back to later, that we have apoptotic bodies, we have cell debris that seems to be protected for long terms of time, and we have leaky blood vessels that would allow a two-way flow with regards to things going into the blood and things coming out of the blood. And as I was saying, hamsters of this age represent mostly what I would consider probably the average teenager. So they don't, our hamsters at this particular age don't succumb to infection. They, like I showed you, they basically plateau in body weight. They probably suffer from respiratory distress. It's based on some food finding experiments. It appears that they lose their sense of smell, but they get all of these symptoms back just like we do. And this corresponds with the generation of spike antibodies in their blood. So their response still works, even though, they did probably suffer for 3 or 4 days. At some point, their immune system wins, very much like your average teenager. However, this is when the story gets even more interesting. So we went back to sequencing both our low MOIs and high MOIs and did more of a systemic look at that same longitudinal study, but looking at various different tissues. So lung and trachea, where we see active virus replication, not surprisingly, we see a robust response to virus infection. So here you're basically looking at interferon-stimulated genes and pro-inflammatory cytokines and anything relating to stress response. And what you can see, I hope you can appreciate, is that at a low MOI, that this response follows the amount of virus there, although it's usually there's a lag by about a day. So peak titer of virus will be here, and then peak response will be the next day. Whereas at a high MOI, these dynamics are lost because initially coming in with such a high MOI, if you remember, we had a plateau of virus levels. And as a result, we also have a plateau of host response genes. So we don't really see a dynamic change that we would see here. And again, I guess you could argue about the relevancy of low versus high MOI, but we focus mostly on low, which has these dynamics. However, the thing I wanted to point out with here is that while we do see virus infection sometimes in the olfactory bulb, there are places where we do not see any virus replication, such as the brain and small intestine, where you are still getting a pretty robust antiviral response, even in the absence of the detection of plaque-forming units or sequencing reads of the virus itself from this sequencing data. And when you take the genes that are differentially expressed between these two, one of the things that I'd like to put out there, and I find quite worrisome, and it is a very active area of study in the lab right now, is that if you go out to day 14 and look at the brain, so if I go back for 1 second. So here in this brain, if you can appreciate the low MOI, so you're settling back down, but there was really no virus in the brain. And I'm sorry for the aficionados out there where the brain is many compartments and this is very crude. This really is whole brain. But what I want you to appreciate is that if you take the whole brain and you look for plaque forming units, you will find none. If you look for reads by deep sequencing, you will find none. But if you do qPCR, which of course is the most sensitive assay of all of these, you can still pick up N, which is clearly, would suggest there's either a low level of virus that's inducing this response, or alternatively, there's something else mediating it. And that's really where I'm trying to get to. But before I move on to that point, what I'm really trying to say here is on this last day, on day 14, I think you can appreciate that we haven't yet gone down to white. So you can see over here in the small intestine, eventually, the response is all but gone, and we've gone back down to baseline with the number of hamsters here. But in the brain, we see a number of things remain, notably translation initiation. So these are all picked out by unbiased genome annotation softwares. We generally use StringDB for this. And if you just take the differentially expressed genes, these are from 6 hamsters that were infected and then recovered after 14 days later versus uninfected hamsters, you can find these 4 very enriched pathways that include translational initiation, mRNA metabolic processing, protein transportation, and nonsense-mediated decay. And if you take those 4 attributes and start plugging them into PubMed and various literature searches, you quickly find that those are the same 4 deficiencies that are enriched in a number of neurodegenerative diseases, including Parkinson's and Alzheimer's. So I'm certainly not ringing any bells or alarms other than to say this definitely requires future study, because it would suggest that there is, in fact, the potential for long-term damage, despite the fact that we don't see any active replication in the brain. And so if that's all true, where does this systemic inflammation come from is the next question I really wanted to address here. And I think I've already started hinting to it with regards to the fact that we really don't see virus infection by plaque assay. So here is QRT-PCR of a subgenomic RNA for the N protein, or the nRNA transcript. And that's relevant because for subgenomic RNA, what we're doing here is we're using a 5' primer that's after that, if you remember the acrobatic polymerase that has to jump to make the subgenomic material. This is a primer that's going to include one end of that subgenomic RNA as well as the other end, which were designed by Matt Freeman, who is a collaborator of ours. And in general, this means that the detection of this transcript would imply that this is coming from an infected cell, because that transcript would not be packaged, and therefore evidence of that transcript should be evidence of replication. And we have certainly been using that for a long time now. And I hope you can appreciate that these are CT scores. And, a CT score of whatever this is, 28 or 27, these are relatively high CT scores and would certainly, you would be deemed positive if you were to test with one of these CT scores. And so here in the olfactory bulb, where we do pick up plaque assays, you can see that we are picking up virus all the way up until day 8. And it's not really until day 14 that this is finally cleared. In the brain, we see the same thing up to day 6, but then by day 8 and day 14, again, virus is cleared even by QRT-PCR, so the most sensitive assay. And as a reminder, this is where we see these neurodegenerative signatures, even though the fact that there's no virus, not even transcript left anymore. And the small intestine, again, where we see no plaque assays, we see virus up to day 4, and then it disappears. And this may, in fact, have more to do with some of the virus that gets swallowed in the process of inoculation, but that's a topic for another day. And so just to reiterate this further, this is a completely independent experiment where we just wanted to address this discrepancy between the ability to pick up nucleoprotein by QRT-PCR and the ability to plaque the virus. And so if you look at the olfactory bulb, so in blue, these are 3 hamsters where we homogenized in PBS, and we either plaqued it or we threw it on Vero cells to see if there was a single infectious unit that would then destroy the culture. And what we found is that there was 1 hamster that could test positive by plaque-forming units, yet all 3 hamsters had a high CT score, which would suggest they tested positive. The same dynamic was true in the small intestine, as well as in the brain, whereas, of course, in the lung, these two things correlate very well. And so, again, we're going to get there, but what I'm really talking about is this discrepancy between the fact that you're getting very high levels of this subgenomic N by qPCR, despite the fact that there doesn't appear to be a single infectious unit in the entire tissue. So ultimately, this comes down to this question is, do you get this level of N by qPCR because, 1 in a million cells are infected, and so it's just such a low rate of infection that you don't ever pick it up by PFU and you need it by qPCR? Or is it that you are generating so much cell debris that you can actually detect some of that cell debris by quantitative PCR, even though there's no infectious virus there? And I'm going to try and answer that question. I'm not sure we have the answer nailed down just yet, but I feel like this is what these kind of talks are for. And so we went a different direction with this, and we took the crystal structure from, it was published in Science a while back this year, and we simply made barcodes on the C-terminal or on the cytoplasmic tail of the SARS-CoV-2 spike protein. So as far as we know, this doesn't have a biological function, and we tried our best not to change any amino acids, and we could create amino acids that now allow us to differentiate viruses at a single virion level. We did this a couple of years back with influenza with much success and could demonstrate where there were bottlenecks between aerosol and contact transmission. But here, we're going to look a little at transmission, but what I really want to get to is what is happening in all these tissues where we don't see plaque-forming units. And so just to orient you on what these graphs look like, so what we're looking at here is every barcode we have that represents more than 1% of the population gets its own color. And then all the other ones under 1% are accounted for in black here. And so what I want you to appreciate is this is our stock of virus. It's noteworthy to tell you that the furin site is intact in all of these tissues. That's important because the loss of furin site in hamsters does result in an attenuated infection. So we started with the stock of virus. We directly infect hamsters, and we have many, many of these. I'm just showing you one representative. But you directly infect a hamster, and then you co-cage that hamster with a naive hamster. And so this contact transmission is very efficient, 100%. But all I want you to appreciate here is that there is no obvious bottleneck between giving a stock virus to the animal and having the animal contacted to a neighboring animal, suggesting that the changes that we've made appear to be of no severe biological relevance, which is an important assumption that we're going to make. Okay, and so this is what the data looks like when we grab, so this is all from one hamster, and again, we have many more, but I'm just showing you a representative. But in the lung, we have this population of variants that have no obvious distinction biologically, but again, allow us to follow virions at a single virion level. And then we just simply look at all these different tissues where we can pick up viral RNA without the ability to pick up a single plaque-forming unit or even a tissue culture infection dose. And what I hope you can appreciate is that while there is some evidence of stochastic bottlenecking, this looks nothing like what it looked like for influenza. And, in future talks, I'll put the two side by side so you can appreciate it. But if this were influenza, what you would generally see is if this were, say, a transmission event, you would see 3 barcodes take over the entire recipient animal or tissue, because that's essentially the bottleneck of getting to that tissue or getting to another animal. But here what we see is basically the entire population of virus is represented in every single tissue, which would suggest to me that one, you must have free-flowing virus. So it's not that you're stochastically killing a few pneumocytes and that edema that we saw was allowing a few virions to get into the bloodstream. And, maybe the vast majority of infectious virus in the serum would be neutralized or destroyed. But every once in a while, in every, in an unfortunate patient, a virus does take foothold in the pancreas, in the heart, in the brain. And that may very well happen and start an infection. But what this would suggest is that the blood, or entry into the blood, or dissemination to other tissues in any other form, is really not a bottleneck, because if it were, we would expect to see individual barcodes dominate distal tissues, which is clearly not what we're seeing here. So this really does suggest that what we're talking about here is access to the entire population throughout the body, which would mean that there is no bottleneck anywhere. That point, taken with this idea that we can't pick up an infectious unit, then would suggest to me that what we really are looking at might just be viral debris, where maybe there's not an infectious unit, but there are products of virus, blebbing apoptotic bodies that have that 80% prevalence of viral RNA that something like qPCR, which is so sensitive, can pick up and detect. And that's why when we detect it by amplicon sequencing here, or by amplification directly in tissues. Sorry. Apparently Microsoft can't handle my presentation. One moment, please.
Too revolutionary what you were showing us.
All right, where was I? Now you're going to see my final conclusion. All right. All right, sorry about that. All right, so now I got my screens mixed up. All right, are we good again? I'll assume yes. All right, so what I'm really suggesting is back to this figure, which I already gave the punchline a while back, is that I wonder, and I'm happy to be corrected, and part of the reason I'm putting this out there is that this maybe is provocative, maybe not, but I'd love to hear some feedback on it. But it's quite clear that based on the data I showed you, based on the successful CRISPR screens that we have performed with Neville, that SARS-CoV-2 infection leads to death of those infected cells. And that death comes in the form of every form of apoptosis, necroapoptosis, pyroapoptosis, and just, your run-of-the-mill apoptosis. And so you're going to get blebbing bodies that have spike on them, blebbing bodies that have RNA in them, and these are going to start floating around in this damaged hyper-inflammatory lung space that includes endemic vessels that this material can get access to. And so my guess would be that small amounts of this material start circulating through your body. Maybe it gets endocytosed through ACE receptors expressing cells, but even this scenario is not necessary. You could certainly envision a tissue-specific macrophage or dendritic cell engulfing some of this material, and then you get that canonical signaling to lead to type 1 and type 3 interferon signaling, because obviously you don't have any of that antagonistic activity happening in this particular cell. And so I think when you look at the signature, so, even in our original paper in ferrets, future papers in hamsters, and COVID cadavers that have been sequenced both by us and by many others out there, you do see an interferon signature. It's not a complete lack of interferon. The problem is that, one, the interferon response is delayed, and two, it doesn't match the amount of cytokines and pro-inflammatory cytokines and chemokines that you're getting. So these two problems are what is creating this systemic inflammation outside of the lung, which has its own problems with all the neutrophils that are there. And so my last little point is just simply about this idea that if everything I'm saying is even partly true, it would suggest that maybe one of the things that keeps SARS-CoV-2 as well contained as it does is in fact the interferon response, in that what we see is, and I should have kept this slide in, but what we see is the upper respiratory tract gets infected. There is a 1-day delay, and then you see a spike in interferon and interferon-stimulated genes. And at that time, the virus moves out of the upper respiratory tract and goes into the lower respiratory tract, obviously to all this naive, fresh cells that aren't ready for it. And so it's clear that the interferon system can protect cells from SARS-CoV-2 if it gets there first. And so you'd have to imagine that the initial response of interferon that's happening in the upper nasal passages may be in some ways responsible for circulating to other tissues. So that systemic interferon that we saw, whether it comes from the initial infection in the upper respiratory tract or some of the debris that I'm proposing is being generated, that interferon signature might actually help keep the virus at bay from other tissues. And if all of that is true, then you would imagine that delivering interferon at the site of infection might actually prove to be either prophylactic and or therapeutic, as long as you put it in the right place, which in this particular case would be the respiratory tract. And so this was the next thing we tried. And, sometimes it feels silly to talk about interferon as a prophylactic because, of course, interferon is going to work against a virus. Interferon is our evolutionarily designed system, but the proof really has to be in the data itself. And to be honest, it doesn't work as well as I would have thought. So this is a pretty typical experiment. So this is 4, 8, 10 hamsters. And you can appreciate that giving interferon intranasally a day before, so this is prophylactically, we can only really drop titers by a log, which is really initially was very disappointing in that you would think that interferon could stop this virus in its tracks, but this virus is definitely still finding ways around it. However, despite the fact that we lose a log measured by plaque-forming units, we lose about the same amount in SGN. So as I was saying earlier, in the lung, we see that qPCR of N levels does correspond nicely with PFU. And we also see a reduction in chemokine. So even though there's a log less, there is something to be said that we see just a diminishment of overall inflammation, which may just be the difference between taking the balance of having the host, run out of control in inflammation versus actually maintaining some level of normal infiltration that's not so damaging in and of itself. And as a proof of principle on that idea, this is just another very typical image, but this is a cross section of the entire lung, and we're just staining for N. So this is the brown that you see here is the nucleoprotein of SARS-CoV-2, and you can appreciate at day 3 with 100 PFU, this virus is all over the lung. So this is peak titers, and there's no way this hamster is feeling very great. But with intranasal interferon given prophylactically, you can appreciate that the virus didn't go away entirely. The virus is still there, but overall we have diminished it. And, I don't actually know why the discrepancy of a 1 log titer would amount to so much less N, but this is pretty consistent with what we've been seeing, that by pathology and by immunohistochemistry, the results for interferon treatment look really good. It makes the reduction in titers very misleading, in my opinion. And just as my one throw out there, that this is not just about hamsters. This is a study that came out in The Lancet. It's an ongoing clinical trial. The reason it's not a Cell Science Nature paper is only because it has a very small number of patients. So this is only comparing 50 patients that received intranasal interferon to 51 patients. These are all people 18 or over. They're randomized. They were controlled well for. There's a good balance between male and female. And these are not severely ill patients. These are patients coming in, testing positive for COVID, and requiring some level of hospitalization. And I hope what you can appreciate is that by recovery, they score their symptoms. And I hope you can appreciate that the administration, this is an interferon alpha 4, I believe, given intranasally, you can see a marked improvement in the patients receiving it, which shows 0 deaths in this particular cohort, where the ones that got placebo, 3 people died. And the only adverse effect upon intranasal interferon was that 10% of this population complained of headaches somewhere during the treatment. So they were not getting interferon, but 15% in this cohort said that they had headaches. So there is a slight prevalence to getting headaches. But overall, there was no talk of depression or hair loss or body aches, which are some of the more nasty adverse effects that are known to occur when interferon is administered sub-Q or intramuscularly, which is the trial that recently failed in Europe. And so with that, I'm going to conclude, and I think these are my 4 major take-home bullets. So the first one is that the response to SARS-CoV-2, as I hope I explained it to you on a molecular level, includes essentially low interferons alongside very high chemokine expression, which is it's a very odd thing. And I think that it is what makes the beta coronaviruses quite unique in what they're doing. Second point I wanted to make is that SARS-CoV-2 infection does lead to robust immune cell infiltration. So, I mean, the pathologist that we're looking at our slides blindly was very impressed to see the number of neutrophils that were infiltrating the lungs. And she mentioned that it was something she had not seen before. Third, the interferon response that is observed, it seems to appear from circulating viral debris. And this is me proposing this right now. Like I said, I'm happy to argue it. Maybe others will find more detailed results. Maybe we did something in error. But thus far, I think the data does suggest that there is either non-productive infections in a very, very few number of cells, or there really is viral debris that may be protected by the nucleoprotein that can then be pulled out with a trizol extraction and test positive by qPCR. And lastly, I just want to again put out this idea out there that it does seem that our own interferon response may control some of the tissue distribution of the virus. This is supported by a recent publication that knocked out STAT2 in hamsters. So they purposely knocked out the interferon response. And there they saw much more viral infiltration into other tissues, again, suggesting that the lack of virus infection that we see might be a product of the fact that interferon got to those tissues before the virus did. And if that's true, then the use of intranasal interferon, either as a prophylactic or as a therapeutic, is something we should definitely consider as a population, both for this particular pandemic, but any future respiratory pandemic that may be to come. And with that, I would be remiss without thanking my people. These guys have been working so hard for a very long time. Ben Nilsson is a postdoc who has been doing all the BSL-3 work. He does drug screening. He lives in the BSL-3. He's currently looking for jobs. So if you're interested, please let him know and reach out. He's especially looking in Europe. Daniel Blanco-Mello, another postdoc who does a lot of the bioinformatics. He is our heavyweight in the bioinformatics department, also looking for a job. Daisy Hoagland, Skylar, and Rasmus are 3 really talented graduate students who all have papers in the works. Marilyn runs the lab. Kohei and Shu are 2 postdocs that both came out of the University of Osaka, and they also have some really exciting work. They did the transmission barcoding work I just showed you. Justin Fair is an MD PhD who's finishing up some really interesting studies about the brain. Alona is our animal technician. Lucia is a new postdoc who's got a whole bunch of projects. It's unclear which way she's going, but all of these people have been fantastic, and I think it's important to mention all of them. And with that, of course, there's lots of collaborators. Randy did our ferret work with Claire, David, and so many other people at Cornell who have helped. Any data that you might want, a lot of it's already posted on Geo. There's much more to come. And feel free to contact me should you have any questions or anything. So with that, I say thanks, and I've really enjoyed the seminar series.
Thank you, Ben. Great talk. We enjoyed your talk as well. And thank you for sharing the new data, and also, most importantly, for putting some very interesting hypotheses for us out there to discuss. Speaking of discussion, let's get right away to audience question. Let's see, we have Mashika. Mashika, would you like to ask your question? If so,
I can see it. I can answer this. So the question is it normal for RNA viruses to take over 80% of RNA composition in the cell? The answer is definitely no. Do you know the mechanism of host RNA degradation? So coronaviruses do a wonderful job at shutting down host translation, and I believe they also degrade RNA. I don't think they approach 80%. I think they're more in the 30% range. We are actively trying to determine why the RNA is being degraded. I think others more qualified than I have suggested that NSP1 itself, that's blocking RNA loading into the ribosome, might also be responsible for its degradation. But I can't say that for certain. So we're looking into it. But great question.
Great. So a little follow-up question here from me. So the level of viral debris and the level of immune infiltration that you see, is that a signature that's kind of unique to SARS-CoV-2 or the SARS family in general, or do you typically see this in other viruses as well?
It's a good question. So I'm a little limited in that the only in vivo work we've really done in my research career has been a little bit of dengue, a lot of flu, and a little bit of VSV, and a lot of now SARS-CoV-2. And out of those 4 models, we've only seen this with SARS-CoV-2. But that's not to say that others have not reported it and maybe I've missed it. I mean, it's a pretty broad, sweeping question if we want to include all viruses. It is kind of interesting though, because labs in general do seem to rely on one assay or the other. And, our lab is also guilty of this, and that it's much easier to do a qRT-PCR where you can use the RNA for other things. And then labs generally rely on qPCR of something like subgenomic N as a proxy for virus replication, which should be a safe assumption given the way that assay works. However, our data, especially the fact that if you take an entire lung, homogenize it in PBS, and then put it onto vero cells, you really only need one infectious unit over the course of 4 days to wipe out that cell culture. And so it's a pretty robust system to look for infectious virus. And yet, time and time again, these are coming up blank. So it really does support this idea that, at least in this case, it is in fact viral RNA that's encapsulated in this N protein. Another point to that, Sarada, and it's a really good question and difficult one to answer, but I think another thing supporting this, which it's hard to show a piece of data to really understand it, but if you look at all of the RNA sequencing data that we've put on Geo, that others have put on Geo, unpublished data, if you align it back to the virus, it's amazing that, let's say you get, 2 million viral reads in it from a tissue. Which is not uncommon. So you get, let's say, 25 million reads of host, and then 2 million of those reads are virus in origin. When you map those back to the virus, I would say 90% of that alignment is only to the N mRNA. And so this virus makes just copious amounts of N mRNA, which means it would also make copious amounts of N protein, which is designed to protect that RNA. So if those cells are dying, there's plenty of protein to go around to grab that RNA. And I would argue that that protein is going to be biased towards binding the actual N mRNA transcript itself, just because it's being translated in the same vicinity of that RNA. And so it's an interesting idea that you might pick up subgenomic N because that's the very thing that's being packaged and spilling out, but that's going to require far more work.
Interesting. Great. A lot of unknowns out there. We really need a lot of more work to understand those. Our next question, let's target Samuel. Would you like me to read out his question?
I can do it. So it says, amazing work and great talk. So thank you, Samuel. Have you seen any SARS-CoV-2 infection of the lung infiltrating immune cells? I would say the short answer to that is no. It seems like infection is largely ACE2 expressing pneumocytes, although the death of ciliated cells would also suggest that there are other cells. We have tried a few single cell from bowel washes, so bronchial alveolar washes, but these turn out to be very difficult because of the edema. You get a lot of hemoglobin in red blood cells, lysed red blood cells in the tissue, even if we perfuse. And so as a result of that, single cell on bowel has been tough. So it's been hard to answer that question. I did ask that question of our pathologist. And if you look at N staining from immunohistochemistry, it's not obvious that there's N staining in cells beyond the airway space that aren't, say, pneumocytes. So I don't think so, but I don't want to give it a hard no. Also, regarding the detectable viral RNA debris in the brain and distal sites, has anyone ever seen similar phenomena? So that was your question as well, Sarada. So I don't think so, but at least not that I know of.
Cool. All right. Our next question is from Maria Paez. This is an interesting question regarding the long haulers. So her question is, do you think that cell debris is also the cause for the long haulers with COVID? And do you see that with hamsters? And is that also the reason why you think some people get it? I'm assuming some people get COVID
Yeah,
infection?
it's a really good question. So we have quite a bit of preliminary data in this space already. I don't want to say too much because it's still a little too preliminary. It's hard to say. So I think that based on some preliminary data, that there might actually be infectious virus in parts of the brain, and I say that only because we've had quite a bit of success detecting both spike and N protein, which would suggest that if it was debris, it would have to be a lot of debris. But that result goes against the inability to pick up plaque-forming units. So I'm not entirely sure where that comes from. But like I said, the lab is very interested in this phenomenon. And we actually have a large colony of hamsters that have already recovered from COVID, but living in the BSL-3. So we are doing longitudinal studies post-recovery to maybe answer some of those questions better. So great question. I don't have a great answer.
Great. All right. Our next question is from Diana. Do hamsters develop any pro-inflammatory cytokines if they don't progress into more severe disease? And if so, does intranasal interferons affect inflammatory markers? I think we'll break this down.
Do hamsters develop any pro-inflammatory cytokines if they don't progress to more severe disease? So we do see pretty strong TNF-alpha signature in hamsters. And there's clearly, based on the lung morphology, there's definitely a lot of disease. There's a lot of dead and dying debris. There are nets. So I don't know that, my guess would be that it just falls on a range where, Akiko Asado suggested this long ago, that basically the more severe cases correlate well with more severe pro-inflammatory cytokines, as well as an interferon response, which would go well with this idea that if you don't control infection for whatever reason, and I think there's probably multiple reasons, that you're going to generate more and more of this inflammatory debris. And hamsters have a capacity to control it like most teenagers would. And maybe that's just that their interferon system is a little bit more sensitive, a little bit more robust, and therefore it can contain the virus long enough for the adaptive response to then neutralize it. Whereas as you age, if anything were to compromise the amount of interferon you can produce, whether it be antibodies to interferon like the Casanova lab showed, whether it be inherent genetic mutations in TLR7, which has been demonstrated, all of these things would be significant factors in allowing the virus to continue to propagate, to continue to make more of that debris, which only will allow more and more circulation. So I think they're all connected, but it's a difficult thing to, again, to pin down. So I'm sorry, I feel like I'm not giving very satisfactory answers to all these great questions, but that's my best guess.
All right, Ben, I'm gonna jump in now. Amazing talk. My brain's kind of blown. Yeah, so when you started talking about the debris, I did some quick reading. And yeah, people are seeing nets everywhere. Unfortunately, there are hallmarks of autoantibodies against like Rho and La.
Yeah.
The, canonical autoimmune, particularly lupus, but also in arthritis, etc. Yeah, I mean, I guess we can't be too surprised by this, but that's not going to be good.
No, exactly.
And then I see that people are it's early days, but they're trying some anti-net therapies, like beads coated in DNase-1, like, mainly used for cystic fibrosis, to try to digest the DNA part of the extracellular gunk.
We're trying this in hamsters with so Rob Schwartz and myself and some of his collaborators have been very interested in nets for some time.
Yeah.
Problem with hamsters is that you first have to make sure that the reagents even work in hamsters and are administered in the same way. So we don't have a DNase coded bead. That would be a nice one because that would for sure be universal. I don't even know the nature of their compound, but they do have a compound that has been shown to inhibit net formation that we are slotted to try in a couple of weeks. So we are going down that route, but we're not quite there yet. Because you're right. I saw her name is escaping me now. Pellegrini she just moved to Weill Cornell, but she was presenting somewhere the other day. I've seen so many talks recently. I'm sorry.
Yes.
But she was presenting at the JAK-STAT meeting that net formation in and of itself can induce interferon. And so if that's in fact true, which does make some sense, like the excretion of double-stranded RNA into the cytoplasm certainly could serve as a PAMP for something like TLR9. And if that was true, then certainly a large part of the interferon signature could also come from dead and dying neutrophils. But that wouldn't explain the inflammation we see in these distal tissues because, there you don't see neutrophils and you don't see net formation. So, I think all of these things are certainly multifactorial. But it's also a really hard thing to study. So we're also trying really hard to try and to demonstrate one way or another if this is true. So one of the directions we've been actively going down is RIP-seq experiments using nucleoprotein pull downs and then sequencing the RNA associated with it to see if it's full length genome or just fragments or even mRNA. So the sgRNA has actually already been spliced. And so that would be really informative. But of course, there's such low amounts, right? We don't even pick it up. So if you look at our RNA sequencing results, even with 25 million reads for one tissue, there's less than 100 viral reads in there. So to immunoprecipitate that material is going to be incredibly difficult. So it's unclear whether or not this will be successful or not, but we are trying.
Okay. That's, okay. So my next one, I'm not necessarily going to fight this battle with you, but I want to pass on that two of our previous speakers have given some data that's highly suggestive that persistent infectious virus lasts. So most recently we had Paul Binash, and if people want to look it up, it's the November 11th talk. He had two, I hope I'm not going to, misrepresent what he said, but my understanding of his data set showed two really salient things. One, they were seeing in fully a third of their patients, they were histologically detecting spike protein in gut after 6 months, which, it could be debris, but that's a long time.
That's a long time,
for debris to last. But then what I thought was the most compelling evidence for a live reservoir of virus is they sampled convalescent plasma from patients in a longitudinal fashion. And then they used that to try to neutralize specific strains of the virus, mostly wild type, and then a combination of two antigenic escape mutations. And they found that initially the patient's convalescent plasma did not neutralize the antigenic escape mutations, but then after 4 to 6 weeks it did, suggesting that in my mind, the only way that makes sense is if there is a pool of virus that is under selective pressure to escape the host antibodies.
Sure. I sadly missed Paul's talk, so I definitely need to go back. And if they're watching Paul, I'm sorry if I don't actually think I'm contradicting Paul. So for one, I'm not saying that can't happen. So if you go to the lung, or the gut, which we haven't done a lot of work in the gut, there could be very small populations of virus. We have this other story that pre-COVID days with Sendai virus, unrelated, but an RNA virus nonetheless. And in my former life, I was very interested in RNA interference. This is actually Schuyler's work as well. But basically, long story short, it's a negative-stranded RNA virus that you can artificially impose RNA interference on by exploiting microRNAs. It doesn't really matter the mechanism, but we can take a Sendai virus that's GFP positive and add it into cell culture and completely destroy it by host microRNA targeting. So basically recreate RNAi. And you can leave those cultures for 5 days and you won't see a single signal of green, suggesting that every virus is wiped out. But if you leave those plates long enough and you keep passing them, eventually, lo and behold, a green spot emerges. And from that, the whole plate gets infected again. And so I am certainly not going to suggest that those kind of things aren't possible, that RNA viruses don't actually have the ability to be at such low levels that they would never be picked up by plaque assay, but might still really be there. I do have a hard time believing that this systemic inflammation that we're seeing would be caused by those kind of events. So I don't think that the observation that Paul is making and what I'm suggesting are actually mutually exclusive in any way.
Absolutely not. Yeah.
There's also this weird thing that happens with measles quite consistently. And again, I'm not saying this happens, but another point to throw out there that measles has this encephalitis form where the genome breaks and the RNA becomes chronically producing the nucleoprotein and some pieces of the genome in a chronic manner. So you're making non-infectious virus in that case, but it happens enough that there is an entire disease associated with it. So RNA viruses certainly can find a way in nature to make their way through it, and I don't doubt that might happen. I actually don't even know what I would prefer, but yeah, that's why I had a hard time with the brain question too, because while we do see a lot of debris, and the debris does seem to be contributing a lot to the inflammation we see systemically, there's also some data suggest that there might be low levels of virus hidden out in very specific parts of the brain, or maybe very specific parts of the GI tract. And maybe that's it, and maybe it's not debris, maybe it's both. And these are the kind of directions we're going now. But I do think we have a much better understanding of, the molecular basis for why you have this massive neutrophil infiltration, why virus can persist as long as it does in the absence of a strong interferon response. And then the edema that we're seeing really does suggest that there is an opportunity for debris and virus to get into the bloodstream and circulate, which would then kind of sow the seeds for inflammation elsewhere. So yeah, I think it's a really interesting place to be, and there's certainly more questions to ask, but I will have to go back and look at Paul's talk more.
Yeah. I just want to clarify that I completely agree that I think the persistent inflammation is from the debris and the nets, and particularly if you start to develop anti-nuclear antibodies, then the neutrophil debris and just then it's a positive feedback loop.
Yeah.
Yes. So also catch Avi Nath's talk on September 8th. He talked specifically about the brain infection pathway.
Oh yeah, I did watch this one. Yes.
Oh, okay. Yeah. Hey, just to remind people, he had some pictures of some pretty wild staining of N and S in brains of patients.
Yes. So we have those too.
Yeah.
And there's definitely infection in the olfactory bulb. That there's no doubt there. So there, it's easy to pick up plaque forming units. It certainly explains why you get anomia with lack of smell. And it does seem to be, there was just a paper in, I want to say Nature Communications or Nature Medicine about that the olfactory bulb, they believe, is the entry point into the brain, which all of that jives very well with what we see, with the exception of, I don't know how long the virus actually persists there. I do think the RNA is there though, for sure. So all great points.
Yeah. Okay. Sarada's got one now.
Yes, you really got me intrigued thinking about this debris and immune infiltration phenomenon. So a very simple question: do you see a sex difference in this phenomenon? I mean, because there's clearly the disease and infection severity has a sex dependence.
That is a very good question. We just got female hamsters in, so we are doing female hamsters studies this week. Although we were not going to address that question, but we absolutely should. So I will get back to you on that one, Sarada, I promise.
Great. And adding to that, do you also see an age difference? Because again, the disease severity also has an age dependence. So do you see this infiltration?
We also, we have a colony of hamsters in a BSL-2 suite that are just there to get old. So we can do those kind of experiments. But as you can imagine, there was a massive purchase of hamsters nationwide. So it's very difficult to get hamsters beyond, I think the oldest we could get was 9 weeks. And you have to grow them out to a year and a half before you start seeing some of the age differences. So we're on our way, but I think those experiments start in January.
All right. We'll wait to hear back from you.
Yeah.
In the meantime, we have another audience question. So let me make sure, are we doing okay with time? Because we are well past our scheduled.
Yeah, I'm okay. I think I have a 1:30 appointment. So
wonderful. So Samuel's question is about, what are your thoughts on other reported receptors like NRP1?
To be honest, if the inflammation is coming from a combination of necroptosis, apoptosis, pyroapoptosis, broken virus, NETs, I think every pattern recognition receptor, regardless of whether or not they are classically viral or not, could be at play here. This is where a model, again, maybe like a Kiko's model, where you can just add an adeno expressing, or whatever it was, AAV expressing ACE2 into some of these knockout mice and then study them, even if it's not specifically in the pneumocytes. It would be a good way at answering those questions. There is this one group that does a few hamster knockouts. So there's a STAT2 hamster animal. But as you can imagine, the reagents for hamsters are few and far between to address some of those questions. But it's a great question. It's very possible, but I don't have a good answer.
Yeah. Okay, Ben. That was amazing. Lot more stuff to worry about now.
Yeah. Maybe hamsters are just hamsters and not people. So.
No, I mean, people are finding the NETs. I mean, it's inhuman,
I know. And there's also there's a lot of reports. I mean, we have a whole center here about post-COVID patients and syndromes of, quote unquote, brain fog and memory issues. And I really hope that it's a minority of patients or only the most severe cases, but it's definitely something that I think the scientific community should now start focusing on if they haven't so already.
Yeah. Yeah. So Ben, do you mind stop sharing? And Matt's going to bring up the slide for next week's talk. So same day, same time. Hopefully everyone can. Oh, I guess Matt's not showing it. Join us next week for, I forget who our speaker is because the slide went away.
Our speaker is actually Neville Sanchana. We had a snapshot of his data in Ben's talk. There we go.
Oh, Neville.
Neville.
Oh, good. Then I'm very glad I didn't talk about the CRISPR screen.
Excellent.
Sounds good. Well, thank you for having me. Appreciate it.
Okay.
Thank you, Ben.
Thanks, Ben. Thanks, everyone. Bye-bye.
Bye.